concept is that enzymes speed up chemical reactions in you by lowering activation energy, it's called. Okay, and if I give a quick example of lighting a match, I think I have a match over here. I do. How do I light it? How do I light the match? I have to take the match and I have to rub it. I don't necessarily have to rub it on here, but I can rub it. This kind of match you can light with a shoe or something, right? Why does that work? Why does the match burn? That stuff on the surface of the match, that red stuff, if I take the rest of the match and do that, nothing happens. Why not? I'm scraping the match on the box and nothing's happening. The wood. Why is nothing happening? Okay, be careful. This is not a living thing. It doesn't use an enzyme. It uses something else called a catalyst. Did you ever learn about catalysts in chemistry? A catalyst is something that speeds up a chemical reaction. A catalyst, now don't write all that. A catalyst speeds up a chemical reaction. Whoops. Speeds up a chemical reaction in a reaction without being consumed. An enzyme is a catalyst. Now hold on. Enzymes speed up chemical reactions. Yeah or no? By doing what the head of this match does. Now all of you are sitting there staring at me when I'm trying to rub the wood of the match on here like you didn't know why it didn't light. Why do I have to strike the red end? There's a chemical on the red end that the heat of friction is enough to get it started. Right? If I take Bruce's paper here and rub it on here, it's not going to light. Why not? That friction is not enough heat. I can show you what enough heat is. If I strike this first, and I have a fire, now if I take Bruce's paper, right, is this going to be enough heat to start on fire? How close? Now, do I have to actually touch the flame to the paper? I can just get it close, right? And what I'm trying to get to is the activation energy of the paper. The amount of heat it will take to start that paper burning. Because that's a chemical reaction, right? What kind of chemical reaction is burning? Endo or exothermic? Exo, it's giving off heat. Right. So the match is like, but not an enzyme. Understand that idea? So I lower the energy of activation. Okay? For example, to break down sugar, I would have to use an enzyme called sucrase to break apart, here's table sugar. You take an enzyme that you have in your digestive system that is used to add water to these two and break that apart. That would be a catabolic enzyme, cutting enzyme. What's so funny, David and Sarah? You sit there with smiles on your face. Okay. Okay, there's a bunch of stuff here about what's called the activation energy, and I think it's best shown with a graph, or the idea. The idea of activation energy is the same idea as this is in your book. It's figure 814. You might want to just jot that down so you know it. Okay? Activation energy is this idea. That in order to get this match to light, the activation energy of this match is what? Whatever the energy of friction is here, right? That's lower than the activation energy of this wood. Does that make sense? Because I can't rub this on here and get it to light. Know what I mean? So because I can put this much energy in to get this to light, then that energy is enough to burn the wood. Yeah? So what enzymes do in you is there's a certain amount of energy it takes to get these bonds to break. We want to take this A is coupled with B, and we want to make A to be paired with C. We want to change these molecules. That takes a certain amount of energy to do that, right? You got to be able to break it and reform it. Right? Understand this idea? If I want to break down this block wall, I'm going to have to put a certain amount of energy into it. This is not enough. I just hit it with my hand, and that's not breaking it down. It would help to focus that energy into a little bitty area and put a lot of mass behind it with a sledgehammer, then it'll break it, right? The activation energy of breaking down the block wall. Does that make any sense? I increase the amount of energy by making a small space and adding a big swing. It's not necessarily a chemical reaction, but the idea is the same. So, every chemical reaction has a certain amount of energy you have to put in to get it started. Enzymes lower the amount of energy it takes. So, an enzyme will make the amount of energy needed to start a reaction lower. Does that make any sense? Okay, now all we're going to do is talk about how they do it. Whatever an enzyme acts on is called its substrate, that word. That's a word you need to know. Whatever an enzyme acts on is called its substrate. So starting now, I'm going to refer to our lab. The enzyme in our lab was called catalase. What was its substrate? The hydrogen peroxide, right? The stuff we put in the graduated cylinder. Still with? If I'm ever going too fast, you just got to stop me and say, I don't get it. So the substrate, our enzyme was catalase, which we got in the potato. And our substrate was hydrogen peroxide. Still with me. So what literally happened, I'm trying to find a diagram just a minute. If this is diagram 817 in your book, this is the enzyme. Enzymes have a specific shape, and that shape corresponds to the shape of the substrate. In this case, the enzyme was to break up hydrogen peroxide into water and oxygen. So the enzyme grabs onto the peroxide. In this case, this one in this picture is doing something different. Grabs onto the peroxide, breaks it apart, and the enzyme is left over to do it again. This part of the enzyme here is called its active site. So the enzyme has a specific shape to fit the peroxide. That's why you can't take spit and make it bubble with peroxide. It doesn't work. You have an enzyme in your spit. It's not the same one. This is why I can drink all the milk I want and have no problems, but my son can't because he doesn't have any lactase. His body doesn't produce lactase, so he can't break down milk sugar. So he has to take a pill with the enzyme to break down the milk sugar. That's what we call, what's the term for something like that? Lactose intolerant. Okay? Questions about enzymes and substrates? Questions about your enzyme, the enzyme in the lab and its substrate? Okay then. Why isn't it letting me pause it? Recording. So, in the enzyme concentration graph, the more enzyme, the more reaction. Right? If I have to cut up 1,000 newspapers, and I have 1 scissors, it's going to take a long time. But if I have 1,000 people with scissors, that's going to go a lot quicker. Okay. Second graph was the one of substrate concentration. Yeah. What the rate is. The rate was how fast that little thing went down and came up. So if it went down and never came back up, the rate is 0. If it goes down and goes up really fast, the rate is the depth, that's the distance it traveled, divided by time. Right? So we have the graduated cylinder, and we measured this distance, let's say it's 10 millimeters, and it took 13 seconds. That's 10 over 13 millimeters per second. That's the rate it traveled at. So, and we said the faster that disc traveled meant that this reaction because when you put the disc in, oxygen bubbles started forming on the filter paper. Because of that the reason for the oxygen bubbles is this is being broken down. Still with me? So the faster this happens, the faster the disc goes down and up. Did I just confuse you? Or do you get it? Okay, so rate, substrate concentration, 0 to 100, well, it's actually 3%, but 3% is as concentrated as we could get. What'd the graph do? Essentially this again. Why? What? Opposite reason. Why is it the opposite reason? Enzymes make this happen, but if you don't have this, you can't make that. Right? If all we have is water, we're not going to make any oxygen bubbles with the enzyme. So far, so good. Let's talk about the last one. I'm going to clear the ink. Temperature. This one to me was the most interesting. Rate. We had 5, 22, 37. By the way, when you're doing a graph, make sure these distances are about the same. This one actually should be over here farther, if that's 0. They shouldn't be equally spaced. Don't forget that. Okay, we did the rate at 5 degrees essentially, right? So what did the graph do? Did it do something like, did it go up like that? Give me some numbers and I'll just kind of mark them and sketch it. What did you have at 5? Give me your number, rate. Somebody just say it out. 4? 1.4. I'll put a dot right here at 1.4. What 22 do, Dave? So about the same. Interesting. What about 37? Anybody else have 0 for 37? You all had 0 for 37. It went down and never came up. Interesting. And 100? Wait a minute. I thought chemical reactions happened faster at hotter temperatures. I thought chemical reactions happened faster at hotter temperatures. Somebody tell me what happened. Becca Olson.
Enzymes, like, they tend to,
Nature.
yeah, they get all, like,
Anybody have eggs this weekend to eat? What kind? I'm a big fan of over easy. Over easy eggs, and you dip your toast in the yolk when you get it all like spread out. Big fan. Scrambled is okay too. Why do the eggs go from a liquid to a solid when you heat them? Shouldn't it go the other way? Proteins have a certain structure, right? So a protein might have an alpha helix shape. And how is it held together? It's held together by hydrogen bonds. The problem with hydrogen bonds, when you heat them, they break. So our nice shaped protein gets all warped and twisted and doesn't work anymore. When you heat an enzyme, the term is denature. And let me go back to our active site picture. Actually, I've probably got a picture of this on this. Just a minute. Let me skip. When you heat an enzyme, if this is its active site, that active site, that enzyme gets all warped and twisted, and the active site is not the same. So it doesn't work. Does that make sense? Can you picture this? Okay. The same thing happens with pH. We didn't measure this. Okay, we didn't measure this. I didn't have you do this part because I can tell you what's going to happen. If we did a graph of pH, it would have done this. We were going to do pH at 4, 7, and 10, and this is the graph we would have gotten. Of rate. Would have peaked at pH of 7. What's 7? Neutral. Check this out though. Your enzyme in your stomach is called pepsin. It works best at a pH of 2. In fact, if you get to neutral, it doesn't work at all. Why not? Same reason. Too much or too little acid can denature an enzyme too. You can make eggs solidify if you add, is it lemon juice to make it work quicker? If you had nutrition with Miss Bachman, you would know. Lemon juice has acid in it. So you mix acid in with the eggs and the protein starts to twist and you get egg white. Oh, cool. So you don't just have to heat an egg to get it to solidify. Although lemon and eggs does not sound good together. Although lemon meringue pie sounds pretty good right now. Even though I'm not really a big fan of pies. had one of the best blueberry pies I've ever had though the other day. Had fresh blueberries in it, not the cooked stuff. Like the blueberries are all fresh, like just yummy. Yes, ma'am. A competitive inhibitor binds to the active site of an enzyme. The reason I'm having you just write these down now is I am not going to ask you to regurgitate these now, but we will use them later. I'm dying to give you a quick example because this is pretty cool. This is how some diet pills work. They bind to the enzymes that would break down your food. No, it all goes out. Non-competitive inhibitors bind to another part of an enzyme, changing its shape so the active site doesn't work. Something that sticks in the active site of an enzyme so that what it normally attaches to doesn't. A non-competitive inhibitor attaches to the enzyme somewhere else, warping the active site. I'll come back to that. Okay, you're going to see the term allosteric site. An allosteric site, actually I'm going to go back to this slide because this slide has allosteric site on it. Oops. This is called an allosteric site of the enzyme, where something can stick in there and make the enzyme not work. Sometimes it sticks in there and makes it work. We'll come back to that too. So active site is where it does the work. Allosteric site is another spot on the enzyme for something to attach to make it work. We'll talk about feedback inhibition later.